Hi everyone. This is a lecture about derivatives of carbohydrates. So we'll see how the carbohydrates and specifically the monosaccharides do undergo some of the reactions. So basically, we know the carbohydrates, and as a particularly if we describe the structure of the monosaccharides, so they are either the aldehydic or the keto polyhydroxyl structure. So they have the major functionality of the aldehyde and the ketone along with the alcoholic functionality. So they can easily go some of the reactions, and we will see how through these reactions they can generate the different derivatives of carbohydrates. So the very first thing is that they uh, go through the oxidation reactions. So the first kind of oxidation of the monosaccharides is that their aldehydic site can oxidize. So for this, the usual reaction conditions are the water medium and the bromine. The presence of these two reagents facilitate the oxidation of this aldehydic functionality. So if the monosaccharides do undergo that kind of oxidation. So this is generating the product which is called the aldonic acid. So aldonic means that aldo is from the this has derived from the aldehyde functionality, and onic is a suffix which we will use for naming that kind of derivative of the carbohydrate once that aldehydic functionality has oxidized. So what the oxidation means is that it has taken another oxygen. So whenever a species is accepting an oxygen, so that is a definition of oxidation, right? So this is not the only definition, but this is a definition of the oxidation, that if a species, an atom is accepting an oxygen, so that is the oxidation of that one, right? So this is generating the aldonic acid, so we usually name it by using the suffix as onic acid. So here we can see the example. This is our starting D-glucose, right? Why I'm saying this is a D-glucose, because this farthest carbon which we see its structure, that this hydroxyl is at the right side if we draw its Fisher projection, right? And that is resembling the D-glyceraldehyde, right? So that's why this is the D-glucose. Now, when that undergoes oxidation, so what is happening, that this functionality is oxidized. So now this is the carboxylate side, which is has converted into from the aldehyde to the carboxylate side. So this is named as the gluconic acid. We are adding the onic acid suffix with the root name of that initial monosaccharide. So this is the gluconic acid. So right now we are not going into the mechanism of these reactions, because that is not the scope of this course. So we will cover the different reactions and how the carbohydrates do what kind of reactions they go through, because they have the functionality. So they give the aldehydic and the ketone reactions, because they have that functionality in their structure. Now, the next kind of oxidation is that if the primary alcohol of that monosaccharide undergo the oxidation, not the aldehydic side, right? So in that scenario, if that functionality has oxidized, so then we will use the suffix, which is the uronic acid, right? So we will use that suffix to the root name of the parent aldose. So we will see how we will name that one. Now, one thing which I just want to refresh for you, that what the mean of the primary alcohol. So primary alcohol means the carbon which has the hydroxyl functionality, and this is only attached with one from the one side to another R group, right? This is the primary alcohol, because this carbon is bonded from the these two sides to the hydrogen, and only from one side this is bonded to another R group, we can say, or another carbon. So this is the hydroxyl function. If there would be another carbon or R group, so then that will call the secondary alcohol. Let's see, the carbon 5 is basically the secondary alcohol, because this carbon is attached from this side to the carbon and to this side to the carbon, right? But the primary alcohol is only the farthest and the last alcoholic carbon which is present in that structure. So if that undergo the oxidation, and that oxidation happen in the medium when we are using the dilute nitric acid, so that go through specifically from this side to the oxidation, and that will be the glucuronic acid. So this is the glucuronic acid, and if the aldehydic side is oxidized, so then we use it onic acid, uronic and onic acid. So you should know the difference between that one. Now another thing is that where we can experience that oxidation of the carbohydrates in our uh, different experiments. So we do conduct the identification experiments in our lab for the carbohydrates. So we do treat with the Fowling solution, Fowling reagent, Benedict, and the Barford test. So these are the tests which involve the oxidation of the carbohydrates. So that's why we say that the carbohydrates are the reducing sugars. So some of the carbohydrate reducing sugar, and when our sugar is a reducing, it means that it is reducing other species, but it get oxidized by itself. So in all of these three tests which we conduct for the carbohydrates identification, so they're basically copper which we are using. So that copper get convert from the +2 to the +1 by accepting the electron, right? So this go through the oxidation reduction reaction, and that is in term of the electron exchange, right? So the acceptance of the electron is basically the reduction, and the removal of the electron is basically the oxidation. So we know that there are the different definitions to explain the oxidation and the reduction phenomena. So that here is to highlight that one, that this oxidation is not only limited to that way only to that reagents, but you can experience in the number of other reactions because of the reducing behavior of these carbohydrates. So they can go through the different oxidation reaction as well. So now the other kind of reaction which it goes is the reduction. So aldoses and ketoses can be reduced under mild conditions. For example, by treating with the sodium borohydrate, which is one of the common reducing agent, to yield the polyhydroxy alcohols. So these are known as aldetols. Again, the ald is from the aldehydic functionality, and etol is a suffix which we will use to name that one if the aldehydic functionality has reduced. Now you have to see that when it get oxidized, so that will convert into the acid one, because with that hydrogen there is another oxygen which has added it to that carbon. So that functionality is converting into the carboxylate side. But once it get reduced, it means that carbonyl oxygen is accepting the proton, and this has reduced that one, right? So in that scenario, this is again having the hydroxyl one. Now this is no more the exactly on the structure of the carbohydrate, but this is now the derivative of the carbohydrate. That carbohydrate has converted into the polyhydroxyl alcohol. So there is no other functionality except the alcoholic side or the hydroxyl group, right? So in that way, we will name it etol. So this is the ribose, which is a 5-member carbohydrate aldehyde base, aldose sugar. So this is converted by the reduction into the D-ribitol. In the next slide, I will show you some of the etols which are the reduction products of the carbohydrate. And I will give you the idea that what are the what is the broad spectrum of their application. So one of this one is the ribitol, and ribitol is a component of the flavin coenzyme, and that is very important coenzyme which is responsible and for assisting many of our biological activities in our biological system. Next is the xylitol. Xylitol is used as a sweetener in sugarless candies and gums. Other one is the glycerol, and there is the myo-inositol. So these both are basically the important lipid components which are also present in our biological system as well, and they are also useful for many of the lipid-based industrial products. Next is the fourth reaction is this one. If we can replace one of the hydroxyl with the hydrogen, so it means we are making that carbohydrate one oxygen deficient. So that's why we use the term as deoxy sugars, means we have eliminated, we have reduced the sugar from its normal count. So one of the very famous is basically the beta D2 deoxyribose. So this 2 deoxyribose sugar is one of is the major component of the DNA. And DNA, you know, this is the genetic material. This is a nucleic acid. So deoxyribonucleic acid. And this is, we know about the structure about the DNA is this is the double stranded helix, right? And this strand is basically made up of this deoxyribose sugar with the phosphate groups, and these two helix are bonded to each other to being in a coiled form to give that structural form to that DNA is that they are bonded through these four nitrogenous bases. So for adenine, this is mentioned as a green shade, and there is a thymine. So they both these are the very complementary to each other. So adenine will only make the hydrogen bond with the thymine, one bigger nitrogen base with one smaller nitrogen base, right? And the cytosine will make the hydrogen bond with the guanine or vice versa. So they are in the hydrogen bonding with each other, and they are basically stabilizing that double helix structure. And the backbone is made up of this deoxyribose with the phosphate unit. Now, the fifth one is that as from the previous slide, if we can replace this hydroxyl group, because why we are emphasizing on the hydroxyl group, because if from the chemistry point of view, if you talk about the organic chemistry and the functionalities and mechanisms, so basically the hydroxyl group is also a good leaving group, right? So that can be replaced with the hydrogen, and that can be replaced with amino group, right? So when we replace this with the amino group, so again this is the deoxy, we will use the deoxy terminology in its proper systematic name, but we will mention also that what is that group which is attached in the replacement of that hydroxyl group, right? So when we have replaced this glucose 1 hydroxyl with amino, so what will be the name of this one? That we know this is the carbon 1, this is carbon 2, this is carbon 3. In glucose, the carbon 3 is having the structure that its hydrogen is on the right side and the hydroxyl is on the left side, and the rest of all hydroxyl are on the right side. We have seen in our previous discussions that in the Haworth projections, we write the right side atoms or the species at the bottom, and which are on the left side, we draw it on the upper side in the ring structure, right? So now this from this you can see that this is a 6-membered structure. This was the aldehydic one, right? And there's a third carbon where the hydrogen is on the right side and hydroxyl is on the left side. So we get that idea that this is the D-glucose, right? And now this hydroxyl has replaced with amino group. So for this, we have to mention the position, and we will use a term to mention that this is now the deficient from the that oxygen. So 1 and 2, this is a 2 amino, 2 deoxy, because that oxygen has replaced from the second carbon hydroxyl group, right? And alpha D glucopyranose. So this is its systematic name. So 2 amino 2 deoxy, that's straightforward. That is to show that this is a second carbon from which we have replaced the hydroxyl with the amino group. Now alpha is for mentioning the anomeric position, which we already have discussed in previous slide, but just to a review, uh, if you just look at this anomeric hydroxyl group and compare the CH2OH position on the carbon 5, so we will see if they are opposite to each other, so then we will use anomeric um, configuration alpha, right? And this is a D gluco. Now this is a 6-membered ring structure, so that's why we will write the pyranose. And if it would be the fifth 5-member ring structure, so then we know that we will use the glucofuranose, right? So that was a systematic name. And in a common terminology, which is also the accepted version to name that one, and this is the a little shorter and the easier one, is that you will write alpha D glucosamine. So amino substituted derivatives are called the osamine, right? So if you will write as a gluco, this is giving you the information that what was the monosaccharide, which has used and its hydroxyl has replaced, and it is giving you the information about the monosaccharide. And osamine is that there is the amino group which has replaced. So this alpha D glucosamine is a common name, but if you have to write its systematic name, then you will write it completely to give the complete information, that is 2 amino 2 deoxy alpha D glucopyranose. And in a similar way, if you just look at this one, if you see the both structures, so maybe some of you are not able to identify that what is the difference between these two structures. So if you look at carefully, so there is the carbon fourth where the position of the hydroxyl has changed as compared to this structure. So now at this hydrogen is at the right side and this hydroxyl is on the left side. So galactose is basically the monosaccharide, and what we know about the glucose and galactose is that they both are the apimers of each other. So the rest of the structure is same, so only the one position hydroxyl on a one position on a one carbon is changed from its the previous structure of the glucose. So the third carbon structure is the same one. So the only change has happened at the fourth carbon. So the fourth carbon change is basically generating its apimer, which is the galactose, right? And if there's a change on the second carbon, so that was the mannose. So these are the three apimers of each other. So now if we name that one, that will be the 2 amino 2 deoxy alpha D galactopyranose, and its common name will be alpha D galactosamine, right? So you should know that you will write the monosaccharide and then osamine for mentioning that there is amino replacement with the hydroxyl one. Now, the sixth point is as they can easily go through the conversion from the linear to ring structures, and there is a functionality. So they also combine and they react with alcohols. So through the covalent bond, and that bond is called the glycosidic bond. So glyco is derived from the Greek language glykos, and it means a sweet, because both most of the sugars were sweet in taste, and when they are generating and making a bond, so that was named as, in nature this is a covalent bond, but in biological terminology this is specific for these carbohydrate linkages, and that is termed as a glycosidic bond, right? So this bond is between, when we treat the carbohydrates with alcohol, so what happens is that this anomeric carbon to the alcohol oxygen is termed as the glycosidic bond. So what will happen here, that this is the anomeric carbon. What was the anomeric carbon? The carbon which is the major functionality carbon, which either is the aldehydic or which is the ketonic one. So when it form in a ring structure, so that get converted into the acetyl or hemiketyl. So this is the carbon one, is the anomeric carbon. So when it combined with the alcohol one, so here in that case, this is the alpha D glucose, and this was aldehydic, so that's where this is the carbon one here, and this is the methanol, which is there to react with that in the acidic condition. So what will be the change here, that this hydroxyl will replace with this methoxy group, right? So basically this hydroxyl will combine with the hydrogen of that alcohol, which is easily removable, and this ethoxy is a good intermediate which can be formed as a good nucleophile, and that will attack on that carbon, right? So I can show you here a little bit about this, so you get that one, because this will capture this hydrogen, right? And that bond will break, and now this oxygen will attack on this anomeric carbon. So now as a result, what we get here, that there is a bond between the carbon and this methoxy, right? And this is called glycosidic bond. So this is the oxygen glycosidic bond, because there is a bond between the O. So you can say this is O glycosidic bond, and general term this is the glycosidic bond. Now it when it react with the methanol, so it can form the two kinds of products. So there could be the methyl alpha D glucoside, there could be methyl beta D glucosides, right? So the product are called the glucosides here, and the bond which is formed is the glucosidic bond. So here, why this is the alpha D glucoside? This has formed the carbon and oxygen. This is a glucoside, right? And the methyl is to mention this methyl here, and the alpha and D is basically because if we just compare their position, so they are opposite to each other, so that's why this is alpha, and D is for mentioning that which is the farthest anomeric farthest chiral alcohol, and how this is its hydroxyl position is on the right side or the left side. So this is the D-glucoside, right? And now here this is the beta one. So beta means that these both are sharing, the heavy groups are both are in the same direction. So that's why this is the beta one. And there is the elimination of the water molecule. So another way, this is also the kind of condensation reaction, because there is a removal of water molecule and that reactant is becoming the part of it. There is another kind of glycosidic bond which is called the N-glycosidic bond, and that is basically present in our nucleic acids. So this is the deoxyribose sugar. This is attached with a phosphate, as I have showed you in your previous slide of the DNA structure. So these are the nitrogenous bases, and this nitrogenous bases are basically combined, and there is a bond between the anomeric carbon and this amino group, right? This nitrogen. So that's why this is termed as the N-glycosidic bond, that this is the bond, glycosidic bond between the carbon and the nitrogen. And if we will write there O glycosidic bond, it means the there is a bond between the carbon and the oxygen of that alcoholic side. So glycosidic bond will also be explained in our next lecture. We will talk about the disaccharides and polysaccharides in a similar way. The more than one monosaccharide units do combine through their anomeric carbons to make the disaccharide bonds. Now, in the next is a task for you. We have nicely discussed the projections that I can show you that this is a way to write the linear structure. To write a carbohydrate, this is a cyclic or a ring structure, but they have their specific particular terms we use for these one names of these projections. So you have to write the projection name for all of these three structures. You have to write the exact systematic name of each of these monosaccharides and the configuration. So configuration means that you have to mention that either that is D or L, right? And secondly, when it is in a ring structure, so either that is alpha or beta. So that alpha and beta is not applying to the linear structures, but for the ring structure, you have to mention that in the anomeric version, either that is the alpha or the beta one. So that you have to submit on a Moodle. If you have any questions, so you can just comment, you can send personally the questions. So I would love to answer you. Have a good day. Thank you very much.